Hello everyone, welcome to the ITLS Academy. Today we'll be discussing on the topic molecular pharma biotechnology. Beginning with the introduction, it is a new form of biotechnology based on through thorough knowledge of DNA molecule and the availability of manipulation technologies of DNA. So, friends, the advances in the determination of RNA structure and function have a significant impact on the drug discovery and obviously on the pharmaceutical industry. This DNA may be the target of the sequence changes in your therapy called gene therapy, or interact with small molecules such as the RNA, the DNA, and the pharmaceuticals, peptides, or large proteins. RNA may be the target for small molecules and proteins for small molecules or interacting large ones. So, the logical and necessary successor to the Human Genome Project, which delivers primary sequence data, are comparable efforts in bioinformatics in order to understand the meaning of the sequences, and also in the structural studies to understand the structure and interactions of the molecules derived from one gene sequence, and the molecular design of interacting small molecules, as well as the overall biological system for validation, verification, and application. So this is the what why we introduce the molecular biotechnology in pharmaceutical industry. The increasing availability of DNA sequences, structural data, and analysis of interaction is opening a plethora of new targets for therapeutic interventions. The application of no we know the application of the genomic technologies to the study of infectious agents is yielding result for anti-infective therapies, and the combination of sequencing, bacterial genomics, bioinformatics, and proteomics provides the targets for the development of the drug, and also to demonstrate the efficiency of genotyping, and to which the susceptibility to infections correlated with the specific gene sequence for any drug development, and definition of a patient subpopulation. Beginning with the first tool as the bioinformatics tool, let me tell you that bioinformatics is an interdisciplinary science that includes the conceptual and practical methods for the understanding and generation, processing, propagation, and integration of biological information. It encompasses the input of the DNA sequences, and it requires the input of the RNA sequences, your protein sequences, and also the structures plus knowledge of the biochemical pathways, your cellular and developmental processes, tissue and organismal physiology, and normal pathological processes. So, the process of designing a new drug using your bioinformatics tools have opened a new area of the research. However, computational techniques assist one in searching drug target and in designing drug in silico, but it takes long time as well as also needs requires a lots of money. In order to design a new drug, one need to follow the following path. That is, he first have to identify the target disease, study the interesting compounds, detection of the molecular basis for the disease. It also requires for the study of rational drug design techniques, and then the refinement of the compounds, quantitative structure activity relationship, and also the solubility of the molecule, and lastly it occurs last step occurs is your drug testing in this field. So it yields knowledge about predicting gene function and structure, protein sequence, three-dimensional structures of the protein and RNAs, and simulations of the gene expression network and of metabolic and signal transduction network. So what happened, whenever we have to design any drug, there are many websites available for the bioinformatics tools in order to design structurally your drug, your structural formula, and your types of the size and type of the structure of the protein particular protein molecule which could be act as a drug. That could be based on computational model. You could correlate each and everything, match your sequences and target your molecule on computer basis on websites. So computational methods are essential in processing the vast data streams from sequencing effort, structural information, protein analysis, and differential gene expression. And computational approaches serves to integrate the research, development, and your discovery phase. Efficiency is achieved by a combination of experiments and computation, that is merging the result of expression profiling with DNA microarray with computation. So, bioinformatics is a key aspect of the drug discovery in the genomic revolution, contributing both to the target discovery as well as your target validation. Next comes your biological systems and models. In research area, your particular biological systems are available on which any drug testing or any drug analysis is performed on that particular model, and then it is considered further for clinical trials. So biological systems are the key to validating the results of the sequencing, structure analysis, and functional predictions. The model employed range from in vitro assay to the cell culture to whole organism. Increasingly, the use of specific differentiated cells provides new model for understanding differentiation and function of the drug discovery and testing, plus transplantation, and as well as also it includes your therapies also. Complex biological systems are defined and studied, which may provide the tool for studying the previously interactable problems. And in the search of the drug to fight tumorigenesis, the study of tumor suppresses gene in human and yeast with its homologous provides such a biological model, and that is an example I'm taking for supporting this biological system and models. We have another model such as your mouse models, and mouse models provide an important system to address studies of the disease development and for the development of testing of the therapeutic compounds and strategies. As why we take the mouse model as somewhat the circulatory system or the blood system of the mouse is correlated with somewhat with the human being. That is why the mouse model is preferably preferred in this biological systems and models for any drug discovery. Next is your assay system. Assay system for the identification of biologically active substance in screening programs, for the optimization of lead structure in the drug discovery, for the test of efficacy, toxicity, and doses need to correspond to the biological target molecule and mechanism. So let me tell you what is a lead structure. In our previous lecture about the drug discovery, do refer to that lecture also regarding the lead optimization and lead structure. Lead structure is that compound that comes in your first phase of your drug development process. In the first phase, first we have to decide, let me remind you, first we have to decide your target molecule, and then the lead structure is decided and the lead optimization and validation occurs. So these were the process in the drug development, and assay system is somewhat related to your that lead structure only. So here what happens, assay system is decided to interact with the lead structure and various tests for its efficacy and toxicity, and what type of dosage should be needed to correspond to the biological target molecule and mechanism are tested here, and a assay system is developed. Validation of the assay system with respect to the biological system for which a pharmacological interventions and therapy is sought in a crucial criteria. So various criteria is also developed in that the that particular chemical compound which is treated to be as a lead structure is being treated and gone through various tests. Signal transduction pathways, receptors, and ion channels are important targets of the assay system due to their amenability to modulation by therapeutically active compounds, their widespread occurrence, and the wide ranging biological effects they produce. So a suitable assay system needs to yield reproducible result and should be amenable to automation. Next comes your high throughput screening. High throughput screening is the description of an array of technologies to speed up the testing of the compounds from various sources, natural or we can take it from the synthetic, for their parameters such as effects with specified test system and ADME. And that is why due to the high pressure on pharmaceutical companies to reduce time to market and to improve the success rate of new drug candidates, higher throughput pharmacokinetics has become an integral and indispensable part of the drug discovery program. And that is why new sample preparation techniques and highly sensitive and selective mass spectrometric detection systems has been contributed to in order to accelerate in pharmacokinetics development, so that it takes less time from drug development that comes from time to reduced time to market. Next is your automation. Automation is required at all stages of the pharmaceuticals, chemicals, as well as biotechnological discovery, development, and manufacturing processes in order to achieve increased speed and throughput. Large central synthesis facilities exist for combinatorial chemistry programs to deliver thousands up to hundreds of thousands of compound for mass screening. At the laboratory level, the preparation of the sample, the dosing of the liquid, and the feeding of the sequencers, your mass spectrometer assay instrument needs to be carried out with minute volumes and with high speed and precisions around the clock. The development of miniatures analytical systems with the integration of sample handling fluids and measurement and calculation is leading to laboratory on a chip devices, and these devices are applicable in laboratory analysis for the development and also in the physician practices. So robotics and automations are applied to the synthesis and liquid handling as well as the detection. The development of miniature analyticals, as I told, it working on your chip devices. So the chip devices system will allow routine diagnostic to be carried out at a genetic and biochemical, pharmacological, and your cellular level, thus enabling the researchers and the physicians to study the SNPs, pharmacogenetics, variation, metabolism of compounds, as well as the genetic predisposition, prenatal diagnostic, and tumor cell profiling on individual patients. Next topic, another tool for this is your genotyping in molecular pharma biotechnology. Genotyping is a method for the sensitive detection of single base mutation in DNA has been described, which involves the application of a primer thiolated or oligonucleotide complementary to the target DNA as far as one base before the mutation site on an electrode or gold quartz piezoelectric crystal. After hybridizing the target DNA, the normal or mutant with this sensing oligonucleotide, the resulting assembly is reacted with the biotinylated nucleotide complementary to the mutation site in the presence of polymerase. The labeled nucleotide is coupled only to the double stranded assembly with the mutant site. The method was applicable to the analysis of polymorphic blood samples with the Tay-Sachs disorder and allows the quantitative analysis of the mutant with no PSR pre-amplification. So here SNP allow us to pinpoint the genes involved in trait and disease, such as we can take the disease of asthma and hypertension, obesity, cancer and diabetes. They will enable both the localization of the gene and gene families as well as the development of personalized medicines. Next tool is your sequencing tool. The gene sequencing has reached an advanced technologies stage where automation allows the rapid sequencing of several 10,000 base pair per machine per day. The future of gene sequencing may be super fast sequencing. So it is a large scale sequencing effort required efficient method of ordering these sequences. Physical mapping has been reintroduced as an important component of the large scale sequencing projects. And another development towards the creation of the ultra fast DNA sequencing is the progress in designing and producing microfluidic arrays. So, this is what the molecular biotechnology has to do with the pharmaceutical industries, and these are the applications where why as how the molecular biotechnology is helpful in the pharmaceutical industries. Thank you for being with us. For more information, you can also log on to our website www.itlsacademy.com. You can also follow us on Instagram, Facebook, WhatsApp, Twitter, LinkedIn, and subscribe our channel ITLS Academy at YouTube.